I'm Jiyoon Han. I'm a third-year graduate student in the Paul Buttress Lab, and I'm a student in gene regulation, epigenomics, and transcriptomics home area in MBIDP. So thank you for giving me this opportunity to present in this great seminar. So today I'm going to talk about my research project, which is about outlier gene expression in cancer. So today, overall, I want to first introduce my project, why I started my project, and what is the goal I want to achieve ultimately. And I developed a new method, so I want to introduce that, and I have some preliminary data using the various cancer data set and want to talk about the future plan. So first, in the introduction, I want to talk about what are cancer outliers and what is the aims I want to know. So before getting into the project, I want to talk about the definition of outlier and what is outlier. So outlier is a data point that differs significantly from other observation. So it's the subgroup of the data points which is far from the other observation. So this is the definition of the outlier, and there is a many type of outliers we could find from our data. So first outlier type of outlier is just an error. So if we imagine that if we measure the temperature in the room, and if one data point is way higher than other data, we could think this is just an error, it's not the real data. So this could be caused by the human error or the instrument error or the data processing error, and there is a lot more kind of the error. And in this case, we'd better remove this outlier point, because if we measure the average temperature in the room, this will distort the real data set. So if the outlier is the error, we'd better remove it. But in the other case, outlier can be the real data, it's just a variability in the measurement. So if we see the one high extreme value, and if this is not an error and it could be just naturally happen, and in this case, it could contain some novelty in the data sets. So here is a definition of the outlier and type of the outlier, and I'm interested in the outlier as an aspect of the variability in the measurement, because in this case, we could know this data point might contain some meaningful or interesting novelty of the whole data set. And then my next question is, so this outlier happen in the cancer? And I want to show some example of the outlier in the cancer. The first example is the gene expression outlier, which is I'm interested in it. I want to introduce the gene, the EML4 ARC, which is a very famous fusion gene found in non-small cell lung cancer patients. It's found in only 3 to 5% of the patient. You could see the small portion of the patient has this fusion gene, and if they have this fusion gene, you can see ARC expression is way higher than the other patients. And this fusion gene is caused by the inversion of the chromosome 2 fragment. So this EML4 is, after inversion, its promoter can activate the ERK transcription, and is activated ERK can activate the downstream pathway. And why this finding is important is this fusion gene is now used for the standard care of the non-small cell lung cancer patients. So they treated an ERK inhibitor for that 3 to 5% of the patients. So this study shows that this cancer gene expression outlier can be some potential driver gene or the potential drug target. And I want to show one more example, which is gene called the GFI1 and GFI1B. This is found in the medulloblastoma, which is a very aggressive pediatric brain cancer. And this medulloblastoma has 4 subtypes, and group 3 and group 4 has a worse outcome. And you can see the subgroup, very small subset of the patients in group 3 and 4 here, they have abnormal extreme GFI1 and GFIB expression. And if those genes are co-expressed with the MYC, they have really poor outcome. And different from the previous fusion gene, they are generated by a bit different mechanism called the enhancer hijacking. So in the normal state, GFI1b and GFI1, they're repressed because they don't have any closed enhancer. But after an inversion or the translocation, they will hijack the local or the distal enhancer. So they could be actively transcribed and make tumor more oncogenic. So in this case, gene expression outlier can be used for the biomarker to estimate the patient's clinical outcome. So the previous study shows that this cancer outlier study and the found the cancer outlier can be some valuable candidate biomarkers or the drug target. But even if we have an extensive amount of the public data, there are a lot of cancer genomics projects, including TCGA. We don't know cancer outlier, any proper cancer outlier detection method can be used in multiple cancer data sets. And so we don't know the number of cancer outliers. So how many there are across cancer types or per patient. Then we don't know, I showed they could be arise from the various alteration, but we don't know how way cancer outliers arise. And we don't know what's their function, what's their role, which function pathway those cancer outliers involved in. And finally, we don't know with the cancer outliers, how they associate with the clinical outcome. So I want to do some comprehensive cancer outlier study using this extensive amount of the public data sets. So first thing I did is I focused on the developing the method. So next I want to show my new detection algorithm. So before getting into the method part, I want to introduce what is gene expression outlier I'm looking for. This is one of the example of the gene expression outlier. This is a one gene, and here's a patient and their like mRNA abundance. And you can see like most of the patient has a almost no mRNA expression of this gene, but very small subset of the patient has an extreme mRNA expression. So this is called outlier I'm looking for. The transcripts that show atypically high or low gene expression in a small percentage of the cancer. So next, how can we detect the outliers? So here is a schematic figure of the benchmarking new algorithm for the outlier detection. So I want to explain each part step by step. So first part is the simulate data generation. I want to generate simulated data to evaluate the new detection algorithm. So to generate it, I use the normalized RNA-seq data, and I try to identify the distribution of each transcript using QQ-plot. And after identification of each distribution, I generate the random number following each distribution to make the negative cases, and I put some outlier cases to make the positive cases. And this simulated data will use for the method evaluation. So after generating simulated data, I developed a new method. And I use 4 different statistical approach for a new outlier detection method. First is mean and standard deviation, and trimmed mean and SD, and median absolute deviation, and the k-mean clustering k=2. So for these 3 methods, I converted FPKM into the Z-score. And for k-mean clustering, I set the k=2, so they will make a 2 cluster. So after conversion, I calculate each parameter. So those 3, I calculate the range of the Z-score, which is maximum Z-score minus minimum Z-score. Because I thought if the transcript has an extreme expression, they might have really high or really low Z-score. So they will have a really large range of the Z-score. And for the k-mean clustering, I calculate the fraction of cluster, which is minimum cluster divided by the sum of a cluster, is same as a total number of patients. And I thought the outliers that will make the small subgroup, so I can calculate the smaller fraction of the cluster, and that will contain the outlier patients there. So after that, I rank them. And these 3 methods, I rank them in the descending order because a larger range has a high probability to be an outlier, And for the first method, I rank them in the ascending order because the smaller fraction has a high probability to be an outlier. So each transcript has a 4 ranks. And finally, I want to quantify the outlier. So first, using rank product, this is way to combine the whole the ranks, and I could get the final outlier score. And then after permutation test, I could get a final outlier profile. So I could know how many outliers there are per cancer and per patient. And here is an example of how my method works. This is a TCGA prostate cancer data sets. And you can see these are like part of the genes, and I calculate the Z-score range of each parameter, mean, median, trimmed mean, and this is a fraction of the K-mean clustering. And even by our eye, this ribosomal protein gene has a really low K-mean fraction, and they have a really high Z-score range from all the method. So this gene has a highest probability to be an outlier, like among these genes. So after that, I calculate the rank, and using the rank product, I can find the top ranked outlier in the whole prostate cancer data set. And these are the top ranked outlier found in prostate cancer data. And I want to double check I really found the gene I'm looking for. So these are top 4 ranked outlier. And you can see it's hard to see this bar, but most of a patient has almost no expression, but only very small subset of a patient has a really, really extreme expression, which means my method detect the outlier properly. So the last part is a benchmarking part, which is I'm working on right now. So I want to test each combination of the method, and using the generated simulate data, I want to calculate the AURC and AUPRC to evaluate each algorithm's performance. And I could select the best performed algorithm. And finally, using that algorithm, I could predict an outlier, and I can apply to the multiple cancer data sets. So this is a whole process of my benchmarking new algorithm. And I still working on the benchmarking part right now, but I checked my method can detect the outliers. So I applied it to some multiple cancer data sets here. So first I tested in the prostate cancer data sets, and I expanded to the whole TCGA data set. So I want to show the first preliminary data I got from the 2 prostate cancer data set. So here are 2 different prostate cancer data set. One is a CPCG, is from the Canadian cohort and has a smaller sample number. And next one is a TCGA prostate cancer data set, which has a more sample number. So this is a 2 different prostate cancer cohort, and I want to know these detected outliers are similar or different. So I want to compare the top 1,000 outliers using my method from 2 different data sets. So here is a top 1,000 outlier and overlap number of the detected outlier. And this is almost 90%, as I was quite surprised, it's quite higher than I thought. It has a p-value equal 0, is really significantly overlap many number of the outliers. And I could see the many interesting gene here, something like ETB family, ETB4 and ETB5, and also ETB1 here. This is a very famous gene in prostate cancer, and known they form the fusion gene with other a famous gene in prostate cancer, and they could be used a biomarker here. So I want to look into this outlier profile later, but to see that their overall pattern I found also interesting. A lot of genes are enriched in the same gene family. Here is a myosin heavy chain, and myosin is an actin-related gene. So these mRNA abundance of outliers belong to the same gene family, which I think these outliers just not showed up randomly. They might relate it to some specific pathway or biological process in the prostate cancer. So I checked their functional analysis of their outliers, And interestingly, most of them are enriched to the muscle developmental cells. So I thought they might be related to some infiltration or invasion to the neighboring tissue. So this will be, I think, interesting study. And also some of them shows that they're related to increasing the heart disease. So also, if you see the overall expression pattern, this is just score metrics of the overlap outliers from the both TCGA and the CPCG data sets. And we could see some interesting pattern. First, the subset of the patient has several same outliers across them, and some outliers tend to be expressed together with other outliers, and some outliers expressed in only a few patients. So we could see they're clustered together. So I will investigate if this group O patient has some specific characteristic, like they have a longer or the shorter survival rate, or they affect some metastasis or recurrence, and also want to see these co-amplified outliers. They might co-amplified from the same chromosomal location, or they might be involved in the same functional pathway. So I want to look into it in the further analysis. So I selected my outlier detection method work well in the prostate cancer data set. So I want to expand in the whole TCGA data set. They have 33 cancer types, and this is a number of the patients, and I will use this figure to know the location of the cancer. So rather than looking into each outlier profile, I want to see more the big picture, how those outlier profiles are different or similar across the cancer types. So I compared the whole top 1,000 ranked outlier across the cancer types. So here is the overlap proportion of the outlier between two cancer types. So these the number shows the overlap proportion, which means 0.05 means 105 outliers overlap between two cancer types. And I use a consensus clustering to cluster the outlier, and I could see some of the interesting subgroup here. First, I see the glioblastoma and lower grade gliomas are clustered together. This really, which makes sense because they are both brain tumor, and it shows that these outliers might really have some cancer specific characteristic, and they're not just showed up randomly. And I could see some more subtype here: prostate, breast, and bladder and lung cancer. I thought they might have some same cancer cell origin, like most of them is from epithelial cells or the gland, which is a carcinoma, and there's a cluster together. And also I could see this as PJAR in stomach and lung cancers are clustered together. And this makes sense because these two are connected organ and they located close. So I think these outliers really contains and have some specific role in the specific cancer types. And I want to show one more data set. And here I want to see the correlation of their outlier score. So I did a Spearman correlation of their top ranked outlier using the outlier score. And we could see more distinct subgroup pattern here. And so first, we could see the colon and rectal cancers has a very high correlation. And we know that colon and rectals are connected tissue. And also, again, as PJAR and stomach cancers are clustered together. And amazingly, they have a really high correlation in glioblastoma and lower grade glioma. And this is adenocortical cancer and the PCPG. Actually, they're from the same organ as adrenal, and inside and outside of the adrenal. And 2 kidney cancer and 2 melanomas are clustered together. So I think this shows the outliers just not randomly showed up in each data set. This outlier really contains some cancer specific characteristic. That's why they have a subcluster together. Also, I want to show one last interesting thing I saw. I keep seeing that cervical cancer and head and neck cancers has a really high correlation and make a subgroup. And we know that human papillomavirus can cause the both cervical and head and neck cancer. So I think those might overlapped outliers might be related to the human papillomavirus, and it could be the interesting study. So these are all the data I have so far and want to show today. And I want to talk about my future plans. And so I'm still working on my benchmarking side. And after finalizing it, I want to apply the whole more the cancer data sets and finalize the cancer, establishing cancer outlier profile. So then I could get the significant outlier profile in each data set and want to identify the clinical association with outlier in each patient. And after the long-term plan, I want to focus on characterizing outliers. So what's their functional pathway they're involved in, or what's their chromosome location, gene length, or gene essentiality. And after that, lastly, I'm interested in the identifying the mechanism of the outliers, which genetic or epigenetic alteration can cause those outlier. And finally, I want to know what is the predominant alteration and regulation mechanism of outliers and how they're different or similar across the cancer types. So this is all I prepared today. And thank you for all the Boutros Lab members, especially Paul and Stefan. And thank you for my committee member, Michael, Heather, and Jason, and Chong Yuan. And thank you for listening to my presentation.
Thank you very much,